Thanks to the discovery of antibiotics, we no longer need to use bloodletting practices like in Bill's time. Now, if you have pneumonia, you would probably see a doctor who would prescribe you antibiotics. For many of us, antibiotics have become commonplace, and you might not think about how they actually fight your pneumonia. But what do antibiotics truly do, and why do we need them? Our bodies have incredible immune systems that are built-in defenses to outside objects and bacteria. Our immune systems encounter and defeat countless bacteria every day without us even knowing or feeling sick. However, some kinds of strong or new bacteria require more attention than others. Our immune systems need some help fighting these kinds of bacteria, and that's where antibiotics come in. Antibiotics are chemicals that fight bacterial infections by targeting processes critical to bacterial survival or by inflicting physical harm on the bacteria. They typically interfere with the bacteria's metabolism, protein synthesis, or RNA synthesis, or damage the bacteria's DNA, cell membrane, or cell wall. This interference can kill the bacteria directly or prevent its ability to reproduce, which allows our immune system cells to catch up with the infection and kill the bacteria. In this way, antibiotics serve as critical assistance to our immune system in treating bacterial infections. In the case of your and Bill's pneumonia, antibiotics can be pivotal. A wide array of bacteria, viruses, and fungi can cause pneumonia in both the community and hospital setting. One of these such bacteria is Acetobacteria baumannii, which is commonly transmitted to patients in hospitals through contact with contaminated surfaces or individuals. A. baumannii cause pneumonia by infiltrating a person's lungs, but they can also cause other infections when they reside in other areas of the body, including blood or urinary tract infections. Thus, antibiotics that fight A. baumannii are critical for patients. Since A. baumannii have protective cell walls, killing them requires stronger than normal antibiotics, such as carbapenems. Carbapenems are a class of more than 80 antibiotics and most commonly treat A. baumannii. Carbapenems kill bacteria with or without cell walls and are used to treat a wide range of infections. However, they are consequently quite aggressive and are generally considered last resort antibiotics for most bacterial infections. Carbapenem's efficacy in treating A. baumannii make them important defenses against pneumonia in addition to other diseases. However, public health experts have identified a growing number of carbapenem-resistant A. baumannii. This means that a large subset of A. baumannii have become resistant to carbapenems, and infections of these kind can no longer be treated by these antibiotics. How did this happen? If you were given antibiotics for pneumonia, or any other sickness for that matter, your doctor probably told you to take the entire course prescribed to you. You might wonder why you can't stop taking them once you start to feel better, instead of finishing the bottle given to you. This is because you can still have bacteria living in you after you stop feeling symptoms. Exposing bacteria to a non-lethal dosage of antibiotics not only means you will probably get sick again soon, but also encourages antibiotic resistance in those bacteria, as they are able to reproduce more and evolve. Like all other living organisms, bacteria often experience variation in their DNA from mutations during reproduction. Most of these mutations are insignificant, and most of those that do have noticeable results cause the bacteria to die prematurely. However, there are some mutations that actually help bacteria live longer by changing the bacteria's ability to survive antibiotics. That's where the survival of the fittest comes in. All bacteria have resistance mechanisms, which are defense systems of their own that are meant to protect from the threats including antibiotics. If a bacteria's genetic mutations occur in the genes that influence the resistance mechanisms, the bacteria could become resistant to certain antibiotics. If these bacteria cannot be killed, an infection of that kind could be incurable. Additionally, the resistant bacteria could then reproduce and spread from one host to others through consumption, bodily fluids, or the environment. Furthermore, antibiotic resistance can even be spread between bacteria in a host body or other environment. Bacteria experience a process called horizontal gene transfer, through which two living bacteria exchange pieces of DNA and thus alter each other's genes. Bacteria can even exchange genes with bacteria of other species. Cross-species genetic exchanges are even more dangerous for the spread of antibiotic resistance, because one resistant species could spread their mutations to many different species of bacteria, making all of them resistant to the antibiotic. In the case of A. baumannii and carbapenems, carbapenem-resistant A. baumannii could spread their resistance mechanisms to any number of other infection-causing bacteria. Thus, growing carbapenem resistance could be devastating for the treatment of not only pneumonia, but also the many other dangerous infections for which carbapenems act as the last line of defense. So if you were prescribed carbapenems for your pneumonia and didn't finish the course, your abomani may become resistant to carbapenem and spread that resistance to other bacteria, both good and bad, in your body. Then, if you later have pneumonia or another infection, you won't be able to be treated with carbapenem. You might be wondering, if antibiotics become useless in treating infections, will we return to a pre-antibiotic world like Bill's? Check out our next video to learn more about how greater use of antibiotics and growing antibiotic resistance might affect our world.